Thank you for joining us for another episode of Biotech and Life Sciences CEO Stories. Today, we're excited to have Carter Cliff of Papillon Therapeutics with us. Papillon's a biotech company that is advancing multi-systemic genetic medicines for the central nervous system disorders. Um, in addition to Papillon, Carter's also founded or co-founded several other companies, including Vascogen, Opsis, Canai, StemCardia, each of which, though, are investigating the efficacy of using pluripotent stem cells in the treatment of various diseases. Interestingly, as a co-founder and chief executive, Carter's raised over $230 million in support of these various programs in obviously a difficult environment. He previously spearheaded the development of a portfolio of pluripotent stem cell therapeutics at Fujifilm Cellular Dynamics. So in addition to talking about the science behind these companies, we're going to spend a little time talking about raising money and what his experiences taught him about that whole often cumbersome process. Carter earned his PhD in molecular biology from UT Austin and did his postdoc work at UT Southwestern. But he now lists several decades in biotech and more than 10 years in the cell and gene therapy space as most relevant to his current work. So Carter, we previously had a guest who was working with small mobile stem cells or SMS cells in the treatment of COPD. Can you explain what pluripotent stem cells are and how they differ in structure and use from other kinds of stem cells?
Yeah, sure. Thanks, Tim. It's great to be here and I appreciate the invite and interest from your audience. The stem cells is a term that gets fairly broadly used, sort of like nanotechnology or other terms that people use for a lot of different meanings. The stem cells that might be most familiar to the medical community would be hematopoietic stem cells, which have been used for nearly a century in bone marrow transplants, for example. And that's the basis for the technology that Papillon is advancing as a spin out of UCSD. The other ventures you mentioned that I had co-founded and advanced developed with iPSC-based technology, induced pluripotent stem cells. And those are a form of stem cell that's earlier that can form any cell type in the body. And so that was a key finding from scientists in Japan and the US, subject of the Nobel Prize some years ago. And the advantage of that technology is that you can engineer and manufacture towards a specific cell lineage, like a dopamine neuron for Parkinson's disease or a photoreceptor neuron for retinal diseases, or a cardiac heart muscle cell for transplant into heart failure patients. And so that's one way of using stem cells to produce a drug product in a controlled scientific way. And then you can manufacture it off the shelf like a drug product. The stem cells that we're working on with Papillon are bone marrow derived stem cells called hematopoietic stem and progenitor cells. And the power of those is to be able to use existing clinical procedures that are well understood and to then gene modify the cells and return them to the patient for a sustained benefit. So just there are quite a few different ways people describe stem cells, and I think those are two of what I view as the most important stem cell products.
So what originally attracted you to stem cell research? How'd you end up in the space?
Yeah, my interest and connection into the space was based on a company that Jamie Thompson at the University of Wisconsin spun out with investors here in this area that were looking to use the induced pluripotent stem cell platform technology to manufacture cells which pharmaceutical companies could use in their drug development efforts. The idea or the vision being that this iPSC technology enabled these companies to unlock human biology. It's not possible to harvest dopamine neurons from individuals. So if you can manufacture something that's approximating that in a laboratory setting and make it commercially viable, then pharma companies, for example, could develop drugs better for that type of cell. But that creates an additional opportunity, which is that degenerative conditions typically are caused or arise or are associated with certain cell lineages that are lost. Photoreceptors, if you lose them, you start to lose vision. For example, if you have a heart attack, you lose a lot of heart muscle tissue, and that's not placeable. So the attraction was that it's a new category of medicine that becomes possible, much like gene therapy enables genetic repair in patients who, have genetic defects that lead to disease. Anatomical repair is needed in patients who have sort of lost tissue function as a result of inherited or acquired disease.
So the idea is to repair rather than attack. Is that right?
Yeah. So the small molecule drugs and biologic drugs that are more commonly known, and I would separate that from biologics like gene therapies or cell replacement therapies. The difference is, at least one difference is that for small molecules to work, for example, to protect the function of certain cells that are degenerating in a patient, the cells still have to be there. So at some point, patients will become unresponsive to these treatments. A good example, again, is Parkinson's disease, where they receive levodopa treatment, but then it becomes unstable over time. And part of the reason for that is that the patients are losing those dopamine neurons in their brain. And so at some point, you need to be able to replace the target cell. And it has become feasible to do that. So it also, portends the question of, when would it be a better therapy to do the cell replacement versus, a palliative small molecule treatment. And so in that particular sector, for example, with dopamine neurons, you see several companies, including one that I co-founded, that are advancing treatments where the dopamine neurons are directly replaced. And in Papillon, as another example, the notion is to replace cells in the bone marrow that form the blood lineages and the monocyte-derived cells that engraft in the tissues. With that approach, you can replace the cells in the marrow in those patients with cells that have the corrected genotype. So then their blood cells and monocytes will have the right protein function.
They correct themselves and regenerate as a corrected cell.
Yeah, they in a bone marrow transplant, you are replacing the patient's bone marrow tissue with new bone marrow tissue. And if you do that with cells from the patient that have been gene corrected, then when you put those cells back in the bone marrow, the new bone marrow engrafts in the patient, that bone marrow will then produce blood cells and a key class of cells called monocytes that permeate the body and deliver the proper therapeutic protein dose to the patient's tissue.
So you mentioned Parkinson's, and I know that, and I wanted to talk about some of the other companies before we really get into Papillon.
Sure.
There's Canine, where you've recently raised, I think, $82 million in a Series A that was led by the Alaska Permanent Fund and Cure Ventures and the Collin Group. So tell us more about Parkinson's and what your work is there and where you are in it.
Sure. Yeah, that story began many years ago when a scientist in New York, Lorenz Studer, and others as well in the field had been focused on using pluripotent stem cell-derived dopamine neurons as a potential therapy. And the colleague that I began working with is Jeff Cordora, who's a scientist at the University of Arizona. And what he did was he took cells that we were manufacturing and placed them in animal models for Parkinson's disease and showed engraftment and durability of response. And so the idea for that therapy really came from a long history going back to the 80s, for example, in which clinicians had been transplanting not manufactured dopamine neurons, but dopamine neurons that were harvested from primary tissues into patients and seeing some benefit, but it was kind of unpredictable, the response patients would receive. The thought being that those patients receiving these grafts were getting a mixture of cells that were impure. And so it was challenging to sort out whether they were, the dopamine neurons were sufficient in that case. But what Jeff Cordor and others, Lorenz Studer, showed was that you could manufacture and get engraftment of manufactured dopamine neurons in a more robust way. And so that's the basis for the lead programs. For example, companies like Blue Rock Therapeutics were launched to develop that. And then under Bayer, that's being advanced in clinical development. Canai is following in that line of development, in that they have developed a iPSC-based dopamine neuron and have published, Jeff has published data showing excellent engraftment and innervation in the animal models. And so now that program is moving into the clinic.
So you're about to go into the clinic with it. That was the point of the Series A.
Yeah, exactly. Mm-hmm.
Terrific. And what's your expectation in terms of a timeline for initial readouts and that sort of thing?
Well, the next step is to file and hopefully receive clearance on the IND. So that's the next step for that program.
Yeah.
Mm-hmm. And then obviously, as is the case for any company, when you're close to that point, you're well along in terms of your planning for the clinical development.
Okay. And then you mentioned Blue Rock and Bayer, and that takes us to OPSIS.
Mm-hmm.
And you fully licensed, I think, that.
Yeah, so OPSIS was formed with a similar goal, which was to manufacture iPSC-based retinal lineages for transplant in patients that have inherited or acquired retinal disease. Common conditions people may be aware of are retinitis pigmentosa, for example, on the inherited side. That's a condition where the concept is to repair the retina by transplanting iPSC-derived retinal cells, photoreceptor neurons. And that work is being led by David Gam at the University of Wisconsin, along with Fujifilm, and they have a collaboration, a research alliance with Blue Rock Therapeutics to bring that program into the clinic. And so that's what OPSIS is focused on. And the idea is the same, which is to replace these fragile neurons in the patients that, when they lose them, they lose a great deal of function, whether it's vision or movement.
So you mentioned, and this brings up a question that fascinates me. You mentioned that the idea is the same. So why use 4 companies to promote the same idea? Why not house it in one? What was your thinking there?
Yeah, I mean, I myself did not have the luxury of doing these build outs from the position of having a fund. So if one is ideating and developing these types of ventures from the position of having an existing fund, then I think that what you're describing is the right approach, and that's what Blue Rock Therapeutics and other ventures have done, is they've collected different assets together, and that has enabled them to have a broader portfolio and a large capitalization. What I have done is work with individual thought leaders and certain strategic partners to launch ventures that do have broad optionality in their specific area, like retinal cell transplantation or dopamine neurons for Parkinson's is just one product of several that could be developed in that area. So I think you make a good point that it is common amongst VC-backed entities and pharma-backed entities to have a large platform potential with many products. That's ideal, just not the position I was in when launching these programs.
But yet, um, I'm going to ask you to be a little self-reflective here. There's something about Carter Cliff that allows you to permeate these various entities,
Mhm.
thought, without without sort of being too self-promotional about it. There's something about you that allows you to work with all these different, very smart, probably very competitive people. What do you think that is?
Um, I think I spend more time trying to connect the dots and enable the thought leaders where I can with respect to company launch, the narrative around the company and its technology and the thought leader. Spend more time on that perhaps than on maybe the sort of helpful self-promotional stuff you could do on social media, for example. I think this is probably the first blog podcast type of thing that I've done.
I looked.
You did, okay. So I guess it's just kind of what interests you, and what interests me is connecting the dots and seeing, where there's potential for something that's very disruptive. And maybe there's not a lot of believers yet, or there's skepticism. So that's a nice place to be. In an ideal world, that would be done from the position of a funded foundry, where you
Right.
sort of create and connect these sorts of ventures and launch them in a more systematic way. So I don't know. It's hard to self-reflect. It's just sort of you just proceed. I mean, it's hard not to be excited by work that people like Dave Gamm or um, Jeff Cordor or Stephanie Shirky, it's or Chuck Murray in the case of the cardiac cells. Like once you start interacting with these experts and their teams, I think just about anybody would kind of get sucked into that.
It it makes me reflect, and I think that the audience will, if they haven't already, by the end of the podcast, say to themselves, "This is a really humble guy,
Hmm.
really
Well,
humble
maybe,
guy." And that, we were talking about the Jacobson book before. That doesn't always happen in the scientific community, though not driven by money. There's a big degree of sort of uh, seeking of recognition there.
mhm.
That your humility allows you to sort of remain in the background and let the bench scientists do their work and let them take the credit. That's what I see.
Yeah, that's probably accurate. It may be a necessary growing pain to become, more self-centered around the financials because every one of these companies needs a lot of capital. But I maybe just that in my case I've had to work through the development from each of the ventures incrementally. The large check from pharma came after many years of development and finding more modest sums of money to develop on or other strategic partners. So that's a different approach than if you are launching, at the outset with a large amount of money. Then I think you have a different task at hand. So
Yeah, okay. And then there's STEM cardia, which of
Mhm.
course is about using iPSC to strengthen and restore the heart muscle and its ability to contract and that sort of thing. And Vascogen,
Mhm.
which is developing iPSC therapies to restore blood
Yeah.
flow, right? Can you share some thoughts about those two, I guess, less developed companies, but what your expectations and how you see the path forward?
Yeah, so Vascogen and StemCardia both share one common thing other than their use of iPSC technology, which is that I think it's fair to say that most of the attention from industry is focused on oncology, followed by central nervous system conditions. And that I'm not sure why exactly that is relative to cardiovascular and vascular. And in the drug industry, obviously now there's a bigger shift happening towards cardiac and cardiometabolic disease. But those companies are developing advanced medicines for treatment of cardio and cardiovascular conditions that are quite prevalent and really problematic. So in the case of Vascogen, we set out to translate technology developed at the University of Indiana, raised some seed funding, and developed a product to a point where it's ready for a clinical trial pending some pivotal animal studies. But that target indication is one in which, there are many tens of thousands of patients in the US annually who receive multiple amputations of the limb because they cannot maintain peripheral blood flow. And that is what Vascogen has set out to do, is to try to address severe ischemic conditions like that. And because the microvasculature is everywhere in the body, there are many such conditions, retinal, kidney, and so on. So that's the idea for that venture. But again, in that sector, cardiac, cardiovascular, it can be more challenging to raise money, for reasons that elude me. For the STEMcardia program, that is a program that industry recognized. Chuck Murray and his team and the amount of data that they had published in small and large animals for their cardiomyocyte transplant product received a good amount of attention from the investment community and was brought into a large entity called Sana Biotechnology, which many people in the space would know. But then, over time, companies like Sana need to refocus, and so that happened. And so STEMcardia is all of that technology and years of effort and a couple $100 million in academic and industry funding to arrive at a product that can now be manufactured at clinically meaningful scale in GMP. And so that's an example of a program that's also targeting the most prevalent serious condition, which is heart failure. It's the number one killer, and globally, not just in the U.S. And yet, as Chuck would describe, the heart is the least regenerative organ of the body, and so you really have to find a way to repair and replace the muscle tissue of the heart in order to help those patients. You may help some of them with some of the newer palliative care, but by and large, pharma have not been able to develop successful small molecule treatments for heart failure. So I think that one also is really on the right track. It just takes time and investment. And actually, yesterday there was an announcement where Chuck Murray and his team will be um, moving, and he's been appointed as head of regenerative medicine at USC in LA, and so that's going to be a good development for the program.
Oh, terrific, terrific. So just to step back for a second, you mentioned the use of pluripotent stem cells in in research for cancer, right? And I saw today there was an article, I think it was in Stat, about Nkarta dropping their cancer work with killer cells and CAR T and going to autoimmune, and that there was something about killer cells that made it especially applicable to autoimmune diseases. Is there some advantage that stem cells might have over CAR T, or am I just
Well,
the non-scientist in me just coming out here?
yeah, I actually don't myself have any currently any projects in oncology. There are certainly some that interest me. And it's where most of the investment in the advanced medicine space, the cell and gene therapy space has been allocated. And I think, to your point, a good reason for that is the circulating cells of the body, these immune cells like T cells and NK cells, for example, are very potent. They just aren't able to penetrate, solid tumors, and they have a hard time attacking cancer cells that are circulating. And so what much of the industry has been focused on are being able to engineer those cells so they are specifically targeted to that patient's class of liquid or solid tumor. And that's one approach that the industry is taking. And so they might, for example, Century Therapeutics is trying to manufacture, as was Fate Therapeutics, pluripotent derived immune cells that attack cancers. So I personally haven't really done much work on that, and I probably would have to defer more to other experts in that area. But I think that the benefit of all of that industry and development for example, what I've been working on and what say Dr. Shirky's been working on is that when we want to manufacture a gene therapy, whether it's for cancer or something else, it builds upon the success of the CAR T or that industry. The way that they manufacture the products, we're able to leverage that. So that's probably where my expertise intersects with what they're doing in oncology, but not so much beyond that.
Okay. So Papillon is using iPSC to address liposomal mitochondrial dysfunctions that underlie various severe diseases like Friedreich's ataxia, which we talked about. But the first iteration of the platform was an indication for cystinosis. And I think you've already achieved a pretty significant level of success with that.
Yeah, and so I'll just provide a summary on that. Stephanie Shirky at UCSD ideated and developed this notion of using the hematopoietic cells in the body to target inherited conditions. She was already an expert in a condition called cystinosis, which is like many of these. It's a degenerative condition that impacts multiple tissues in the body, and so it's a difficult kind of protein to target with traditional drug development or gene therapy because it's transmembrane protein that's kind of stuck in the membranes of the cell, and so it's difficult to target it. Additionally, most of the small molecule and gene therapies, for example, would often be targeted to one organ, like the heart or the liver or something like that. And it's more commonly the case that patients who are afflicted with these kind of conditions, these inherited conditions, it's not just one part of their body that's affected. So the cystinosis patients accumulate crystals, cystine crystals in their body in many tissues, and some tissues are affected more than others. But it's overall a condition where, in an ideal situation, you would be able to leverage the capacity that the body already has to deliver cargo to different tissues. And different groups have tried to do that sort of thing using circulating cells like blood cells. But what Dr. Shirky learned and her innovation was to apply a gene therapy approach to cystinosis in which the gene therapy was delivered via the hematopoietic cell transplant. And those cells with their corrected protein would then have daughter cells that are monocytes, which can go into all of the different tissues of the body and deliver the protein. And so she carried that from ideation in the lab, as they say, bench to bedside, all the way through the conclusion of a successful phase 1/2 trial last year. And at that point, Novartis elected to take the program over and is carrying it forward. But the results of that trial for many of us in the field were profound because, for example, the patients, by and large, do not need to take this medication, which they would have to take, 30, 60 pills a day, a large number of pills, just to maintain the, a kind of safer level of cysteine in their body. So that's pretty profound for a first in human safety trial. And I think that is, for many people, what really interested them and myself included in what she's advancing with the hematopoietic cell gene therapy. And that's the basis for Papillon's pipeline.
And just to give her an appropriate amount of credit, I think she's your co-founder there as well, right?
Well, she and Eric Adler at UCSD and Farah Sheikh and Jeff Ostrov are the co-founders and sort of scientific co-founders. The whole platform technology and IP is really based on what Dr. Shirky has developed. And then there are specific applications such as Friedreich ataxia as a target indication and Danon disease as a target indication where the other founders have specific expertise. So it's a group of multiple founders who have spun the company out of UCSD. And then my role coming into it is to ensure that it's able to capitalize and develop, and that there's a kind of commercial strategy to the pipeline.
Okay. So let's talk about Friedreich's ataxia for a minute. What is it? And really, why am I seeing it being targeted by so many companies?
Yeah. Let me go with the latter question first. So Friedreich ataxia, which I'll describe in a moment, it's a condition that where the patient community has a very, very good disease advocacy foundation. It's called the Friedreich Ataxia Research Alliance, or FARA. And the management of FARA have done a really amazing job of organizing that patient community and organizing the biotech companies that are interested to develop treatments for it. And, for example, to conduct what are called natural history studies in those patient populations so that it's more straightforward for companies to develop treatments. So that's I think one reason why it's an area that there's a robust pipeline of therapeutic programs for. Last year, the first small molecule for Friedreich ataxia was approved by the FDA by a company called Reata. It's called Skyclarys is the name of the product. And that company and product were acquired by Biogen, who's now marketing it. And behind that approved small molecule, there's a sizable pipeline of different types of therapeutics, of different modalities that are being developed, of which our approach is one. So I think that's great news for that patient community. And about the disease, so Friedreich ataxia is a disease caused by what's known as a repeat expansion in front of a gene called frataxin. And a repeat expansion is where the gene itself may not be mutated, but there's a region in front of the gene where there's a repeat of GAA, and everybody has this repeat, and it's related to the regulation of expression of the gene. So it determines how much of the protein is in the tissues. Okay. And in the patients who suffer from Friedreich ataxia, they have way too many of these repeats. And the number of repeats those patients have is variable, but it's a little bit like conditions such as Huntington's, where you have a repeat expansion as well. And the degree to which the repeat expansion is excessive, has an impact on the onset and severity of the condition. And so, like many of the inherited conditions, Friedreich ataxia has degeneration of multiple tissues in the body, but notably, one tissue that degenerates is the nervous system, the central nervous system. And so the patients, are not necessarily losing cognitive function, but they lose motor function. So they eventually need to be in wheelchairs and they can't move very well. But then it has an additional impact, for example, on the heart, where the patients, as a result of the degeneration, end up with a cardiomyopathy that is typically what would be causing, the morbidity of the disease. And so it's a great example, along with the other examples of cystinosis and Danon disease and so on, where of course, it's a great benefit that there are gene therapies or small molecules in development targeted to certain tissues like the heart or the central nervous system. But ideally, what you want is something that is going to be able to deliver a treatment to multiple tissues of the body in parallel. And that's where Dr. Shirky's innovation comes in and where Papillon has sort of differentiated from others in the space.
Okay. That's what makes it different from Lexeo, everyone else in terms of approach.
Yeah, it's Lexeo and others have great programs that are using AAV, which is a virus, to deliver the protein, sort of add the protein to the tissue is maybe one way of thinking of it. And that makes sense. That makes a lot of sense to develop that type of approach, and they have been very successful along the way. And what's different about what Papillon's approach is just that there's a gene correction to restore the normal genetic promoter region, or this repeat expansion, to its kind of normal level. And so you're not necessarily adding the protein back to a specific tissue, but rather you are correcting the protein to its normal expression level and then enabling the body to deliver that via the hematopoietic stem cells in the bone marrow throughout the body, rather than, for example, with a virus to the heart or to the central nervous system.
Okay, so it's broader. Where are you in terms of the clinic and timeline and that sort of on Friedreich's ataxia?
Yeah, so the program is fortunate to have funding from the California Institute for Regenerative Medicine, and it's a preclinical stage program. So it is at the moment conducting what are called CMC and manufacturing, as well as IND enabling animal studies. So it's, at this point, it's going to be, a few years before it's available potentially for a clinical trial. But that's really obviously what the goal is and what everybody at the company is focused on.
Okay. And then Dr. Shirky also is applying the same techniques, I think, to Alzheimer's.
That's right. Yeah. So she has published recently a paper illustrating what I find to be very compelling data, showing that if you intervene in animal models of Alzheimer's disease with hematopoietic cells, you can sort of prevent this pathological and neuroinflammatory environment that is thought to be a cause/symptom of the disease condition. And so the idea is that whereas most groups are very focused, for example, on finding molecules that can disrupt the plaques that may appear in that kind of neurodegenerative condition in the brain, what her approach is to try to address the underlying sort of autoimmune or inflammatory condition that may be leading to or resulting from those plaques. And the hematopoietic system is the natural vehicle or tool with which to do that. And so probably the best thing would be for viewers, to look at her paper. And it's quite clear when you look at the data that, in animals that are control animals that are not the Alzheimer's developing model, you don't see this plaque formation. And then if you have the untreated animal models, which develop the plaques, that's quite obvious from the histology and other data. And then if you deploy the treatment that she's evaluating in that model, it reverts it back to the non-Alzheimer's phenotype quite clearly. So that's usually the sort of thing that groups do to establish the potential of their treatment. And so that is a natural target indication. The question that's being investigated now is really what sort of gene modifications to the hematopoietic cells would make sense for which sub-indications of Alzheimer's disease, for example. And so that's where the company and her program they're still developing that.
Okay. Papillon has a kind of an interesting corporate structure, virtual corporate structure. Can you tell us about that and why you chose to go that way?
Yeah, that is the capital efficient way to develop early on. And a good reason to do that is you can work with the non-dilutive funding, for example, that's available through the California Institute for Regenerative Medicine, CIRM, through NIH, et cetera, and basically utilize that money to advance the programs such that they're closer to clinical development, which makes the investors more interested in the program and makes the pharma more interested in it. So that's really the logical approach for developing, at least in my perspective, these sorts of therapies. And fortunately, the pharma make that feasible by having incubators like J-Labs, which is where Papillon is sited outside of San Diego in the J-Labs facility there, where you can still do the research and development within the company, and you can be adjacent to the research institution where the scientific founders are based. So to me, that's an advantageous setup, and it's more straightforward than spending substantial amount of capital to do kind of bespoke build out for your research and development. So it's really just a question of kind of risk and stage. And for Papillon, as an example, that's really the right approach. And it is, for me anyway, just kind of the standard playbook because when you're operating with seed financing, initially you don't want to sort of spend your capital on infrastructure as much as you do on the programs themselves.
Proving out the potential validity. So
Yeah.
Also in terms of structure, and we've already talked about these separate companies, all using essentially the same platform. Are they sharing things like administrative costs and things like that in order to kind of winnow down what those expenses might be? Or
Not at the moment, no. They're all separate entities that operate separately, but again, the capital efficient way to do that is to to outsource a number of those functions. And so in a way, by having a virtual structure with, for example, sited at J-Labs in San Diego across from Dr. Shirky's labs at UCSD, we can work with groups that can handle the different functions of a biotech. We can outsource the accounting and we can have fractional executives helping us on the finance side and so on. And so that's all separate. In a venture fund that may have a studio, they would consolidate those kind of activities and they would consolidate management teams and so on. So that may be a better model. It's just not a model that I have access to at the moment.
Okay. So Carter, to circle back a little, I think you've raised $230 million, give or take, in all these various ventures. Can you talk a little bit about your experience in terms of accessing financing? Been pretty successful with it.
Yeah, I mean, it depends how you define successful.
There's still so many aren't. So yeah. I turned that successful.
Yeah, I find the most satisfying moment to be when you raise your initial seed round, because that's when you really get some momentum 4-year program, where you can start to make important tactical and strategic decisions about the pipeline and about the company build and that sort of thing. And so for me, I have more connections with the thought leaders and the scientific experts, and I needed to build out connections with the investment community. I had the opportunity to do that when we initially went out to raise funds for OPSIS, sort of going back to 2016, 17, that kind of time frame. And at that time, what we did was we did a parallel effort with venture capital groups and with pharma, and it kind of just shook out that the pharma interest in the retinal cell transplant product was quite high. And so we ended up going with a pharma transaction for that program. Looking further along to, for example, for Kenai, which had a different name before, but when we were launching that company, we worked with a fund and raised a smaller amount of seed capital and developed the program towards an IND filing. And that's the point at which there was interest in the last, let's say 18 months or so that it took to talk to different groups in the venture capital community. That's where the interest arose for that program, which is a reflection of kind of the reset of the market towards, less early stage stuff and more clinical ready programs. So that takes quite a while. I mean, it takes, whether you're talking to pharma or the venture capital community, it seems a couple of years to do that really. For Papillon, it's similar. We are raising seed financing for StemCardia. They're similarly raising, seed financing, and the purpose of these initial rounds is really just to kind of kick off the strategy for the company, right? And for those sorts of raises, it makes sense, at least in my mind, to connect with members of the investment community who know the space very well. Like maybe they know Friedreich ataxia or cystinosis quite well for various reasons, and they have a good understanding of the financing process, through venture or pharma following that. And so in Papillon, for example, some of the investors are founders of hedge and private equity funds, disease foundations, that sort of thing. And so they're familiar with the opportunities in that space. They're familiar with cell and gene therapy. They're familiar with the disease conditions, and so they think Papillon's a good bet for those reasons. And each time one goes out to raise funds, it seems like it's sort of a different experience, at least for me.
Yeah. But the two themes that I hear that you've focused on is, one, you have a different approach. You're not a me too. And then two, to focus on financing sources which have some adjacency to what you're doing. It's not out of the blue, which requires some diligence, I would think, initially. It's not just off the top of your head. Was there a process to learning how to raise money? Or did you just kind of throw yourself into it and learn by mistakes?
Entirely the latter. I don't know. I know that there are a variety of ways, apps, et cetera, in which there's, probably within that, there's some way to do financing in a more systematic way. But each round, each venture, the deals they secure are unique, right? And so I think it's purely empirical, at least for me. I believe though that, again, if I were in a different position where I had let's say a funded foundry or a fund of some kind, then I think you can be more systematic, because you're doing the company building and the diligence and ideation and so on internally with the knowledge that you're going to be able to sub-select from those and launch or de-risk ventures. So for me, I have not been in that position as of yet. and therefore needed to treat each project, at each venture and company as its own kind of individual that had its own needs, really. So it'd be nice if there was a systematic way to raise early stage funding, but I'm not sure there is.
Yeah, well, there were a book.
Yeah.
I think that when we first talked, you said that most of what you've done has been through angels and high net worth um, family offices, things like that, rather than VCs. Do you think is there a difference in the way that you approach one versus the other, at least as far as you're concerned?
Well, I mean, one obvious difference is that, for most venture funds, they want to write large checks. So you need to be ready to have a use of capital that reflects that. It's not true for all of them. Some want to write smaller checks, but for something like an advanced medicine, which is going to require a lot of capital long term, you need to have a meaningful ask around that. I don't think it makes a huge amount of sense to focus heavily on the venture community until you are ready to reach an inflection. So you need to be, at this point anyway, you need to be closer to IND. The exception for that seems to be if there's some particular, institution or thought leader or angle that a fund is interested in already, and then they may launch something that's more of a platform with earlier stage programs. But that seems to be less common now as the risk profile has changed.
Okay.
So with the angel community or with offices or other strategics that are not venture, I mean, I think the important thing is you're not raising too much too early, because they don't want to, suffer too much dilution before you create value. And that you don't have an unreasonable, valuation expectation, I guess, or seems sort of most common. But I think you also just want to be talking to groups where you can identify there's an overlap. Maybe they're impact investors, maybe it's a larger company that has a corporate fund that'll co-invest, but it needs to be aligned with something that's of interest to their company.
Right.
So those are the kind of things you would look for.
Right. Okay. Carter, what do you love most about building companies?
I think that the most satisfying part of it, as I mentioned, is once you have raised your initial meaningful amount of capital with which to advance the company, because then you have taken something that was embedded, for example, in an academic center, and set it on its path to translation and potential, benefit to patients and so on. And for me, that's intellectually the most interesting inflection, is that moment. But I think for it will also be very satisfying, I suppose. For me, it's not so much the company building, it's more about the programs. Where are they at? Are they, so it'll be very satisfying for me to see, a positive clinical trial with the retinal cells, for example, or a positive clinical trial with the Parkinson's cells, or additional positive clinical data with the cardiac cells, or Stephanie's hematopoietic gene modified cells. Those, I think, are where the decision early on to align and work with each of these groups makes the most sense once you get there. I personally think, for example, that once the iPSC photoreceptor progenitors are transplanted into patients and they are allowed to mature for some time, I think those patients, there's a good chance those patients will have profound improvements, in their vision. And so for me, that would be sort of the watershed, satisfying moment for having worked on all of this for so long.
Having made a contribution, that's what it's about.
Yeah, just seeing the result that actually, takes the innovations from the thought leaders and scientists and shows translation of them. And once you have early clinical data where there's some meaningful signals of efficacy, it's certainly not a straightforward path from there onwards. But these kind of treatments, they're different. They have a higher probability once they've proven out in terms of safety and early efficacy. They have a higher probability of reaching commercial. So I guess phase 1-2 readout, early efficacy in a clinical trial is kind of what is of greatest interest to me to see through.
Right. And commercial for you means someone can see, right?
Well, I guess what I meant is when a product is actually on the market. But I think that, I don't think there are any of these programs which, if they're showing that type of profound effect on the patient, I'm pretty sure by then, pharma or some other company would have acquired them.
Yeah. Okay. Well, Carter, I'd like to thank you so much for taking some of your time out of the day and spending some time sharing some of your thoughts with me and the audience, obviously. I wish you all the luck in the world. I'm sure you're going to be successful with at least one of these ventures. And I applaud your humility.
Okay, thanks, Tim. Yeah, I really appreciate it. And it was great to be here.
Thanks for tuning in to the Life Sciences and Biotech podcast. We'll see you in the next episode. The information contained in this website and podcast are purely informational and not considered investment recommendations. Tim Doherty's participation in Biotech Insights is separate and apart from his role as an investment advisor representative. Nothing contained herein can be construed as a recommendation or endorsement of any of the companies discussed. Tim Doherty also has no financial affiliation with any of the companies mentioned in this communication. Tim Doherty makes no representation that the information contained in this material is accurate and is under no obligation to update this information as changes occur.